Hello students. Continue with the chapter Human Reproduction. Today we are going to study post-fertilization changes, that is, we will start with embryology in humans. The development of zygote and formation of embryo, and after that differentiation, that is formation of different organs. So in most of the animals, these stages occur during the zygote development, that is after fertilization. The first is embryo. The first is zygote develops and it divides rapidly through, of course, mitotic division. But this type of division, the cleavage is slightly different from mitosis. We will see in the coming slide. Early cleavage stages, okay, where it will form the solid ball-like structure, that is morula, and after that it will form the blastula stage with the cavity blastocele. After that, the blastula gets implanted on the wall of the uterus, and then the next stage is followed, which is known as gastrulation, where the organogenesis occurs, because first the establishment of three germinal layer, that is ectoderm, mesoderm and endoderm, and then these three germinal layers will form the different organs in our body. So the next step will be, of course, the body plan, where the neurulation, that is the neural plate, is formed, and then all the organs formation followed by neurulation. So the first will start with the first event, which is early cleavage stages. See, when the zygote is formed, after that, after 24 to 30 hours, the first division occurs in the zygote. This first division which occurs is a rapid mitotic division which starts, and this type of rapid mitotic division is known as cleavage. Okay, so the first division in human which occurs after 30 hours, this should be the meridional plane. Okay, so it will divide into, this is the first division, so it will divide into two cells. Then the two cells will form four cells. Okay, the two cells will form four cells. I'm drawing different pattern because different pattern of cleavage is there. I'll discuss with you one by one. So cleavage is, then this is actually 40 hours when the second division, right? Then 72 hours. Okay, so this way the third division, and then the cells which are formed, they multicellular structures, that is 8 cells to 16 cell stage, and after that 32 cell stage. Okay, once the division starts, then it occurs quickly. Okay, so cleavage is different from mitosis, because in cleavage actually what is happening, the size of the zygote, that is the embryo, remains same and division is going on continuously. The number of cells are increasing, but the size of embryo is not increasing. Of course, the cleavage is it's a sort of cytokinesis only, that is the division of cells. It's just not nuclear division. Division of cell is there complete, but the size of this embryo is not increasing. Not increasing the size. This is actually cleavage. Okay, now this cleavage is very much important. This cleavage is important. What is the importance of cleavage? First one, the zygote can undergo differentiation, and it can form large number of cells, so that the organ formation can be started at the time of gastrulation. So large number of cells can be formed first before organization. Okay. Second one, so large number of cells can be formed. large number of cells formed. Second one, it keeps high nucleocytoplasmic ratio. So high nucleocytoplasmic ratio, it keeps high nucleocytoplasmic ratio. Why? Because eggs actually need a lot of cytoplasm to support embryogenesis, and it is difficult or just impossible for one nucleus to support a huge cytoplasm, and oocytes are the one small nucleus, and the large cells cannot do that. So the more number of cells are there, so the larger nucleus, larger number of nuclei are present, and that can easily, so the size of the cell is small and nucleus is large. Okay, one more difference, that is third one, that is this cleavage. When this type of division occur, here the G1 and G2, it is of very short duration. G1 and G2, very short duration. So size is not increasing, and G1 and G2, size of embryo same. Actually, these are the two differences in cleavage and mitosis, and the importance is large number of cells can be formed and keeping high nucleocytoplasmic ratio. Now, the cleavage pattern we are going to discuss, that what type of cleavage pattern in human is. Okay, the cleavage, various pattern of cleavage, it is based on first basis, right? Pattern of cleavage we are discussing. So first one, on the basis of furrow formation, on the basis of division, furrow formation. Now furrow formation, that is if the cleavage furrow extends completely, and this furrow, this is furrow formation, which divides the zygote into two cells. This furrow is dividing complete zygote into two cells, right? Then that type of holoblastic. So that type of cleavage is holoblastic, complete furrow formation, which will result in two cells. And meroblastic. Meroblastic means if the yolk is present in the egg. Holoblastic example we can write humans, because there is no yolk, so the zygote is completely dividing, okay, fertilized egg. But meroblastic, where the yolk is present, then the cleavage will be restricted to the germinal disc, and the yolk part will remain uncleaved. Okay, so maybe the yolk is present here, then only the cleavage is there. Cleavage will occur in the portion where the egg cytoplasm is there. So the zygote is not completely divided, because here the yolk is present and here is the egg cytoplasm is there. So okay, so meroblastic, where the cleavage is restricted to the germinal disc only, which will form the embryo, and the yolk will remain unaffected. So division will occur here only, and that will result in the cells, the growing cells will be formed here. What will happen here? The complete division. The complete division will be there. Okay. So the first one is holoblastic, meroblastic, based on the formation of furrow. It is complete, then holoblastic. If it is incomplete, it is incompletely divided the zygote, it is meroblastic. The second way of cleavage pattern, that is on the basis of fate of germinal layer. When the cleavage occurs, and in the later stages, if the animal is diploblastic or triploblastic, based on that, the embryo will form either 2 germinal layer or 3 germinal layer. Okay, so when the germinal layers are formed, before that, if like this is the zygote, the cleavage is occurring. Okay. Now, these two cells will form 4 cells, of course. 4 cells will form like 8 cells. Okay. Now, in this condition, if one part, A and B, right, these two parts get separated, right, then these two parts will form the complete organism on their own. Complete organism can be formed from A part also. Complete organisms can be formed from B part also. What does it mean? Initially, the cells which are formed, they are totipotent and they can be differentiated into any kind. They can form any type of organ. That is known as actually indeterminate cleavage. Indeterminate. Indeterminate. In humans, indeterminate type of cleavage is there. And till blastula stage, even before gastrulation, when the organogenesis has not occurred, up to that time, the cells which are present, they are totipotent. You must have heard the term stem cells. Okay. So in humans, indeterminate. If the early embryo is broken down, then all the cells have the capability to form the complete organism. Indeterminate. Opposite to that, the determinate type. What is determinate type? Determinate type means if this zygote is dividing into 2 cells and A and B, these cells will get separated. Neither A nor B will be able to form complete organism. Complete organism cannot be formed from separated cell. That is known as, what does it mean? These blastomeres, after cleavage, the cells which are formed, they are known as blastomeres. These blastomeres have predetermined future. Predetermined future. They are going to form, since, in the beginning, they have the future that they are going to form a specific organ. And that is why it is known as determinate. Okay. Now the third type of, so we have, till now we have done 2 types of classification based on the furrow formation and both based on the fate of the cells. Now the third type of classification, on the basis of the cells, blastomeres, which are formed on the basis of their arrangement, whether they are, radially arranged, whether they are spirally arranged, or they are rotationally arranged. So, radial, spiral and rotational, 3 types. Radial means the successive cleavage, they cut us straight through the egg at right angles to one another so that the resultant blastomeres will be arranged radially. What does it mean? Here, the complete radius, then this right angle, this one, then the radial, this one. Okay, so the radial arrangement, when this is there, then this one. So the complete, this type of division is arrangement of the blastomeres. If they are right angle to each other, then it is known as radial. Spiral. In mitotic spindle of the 3rd cleavage, okay, the 4th blastomeres actually are laid down obliquely. So what is actually here, after the outer layer, the inner quarter or inner group of cells, they are embedded inside. Like for example, here is the inner one, then this is the outer one. The division actually occurs spirally. So the interior tire is actually embedded in the with the exterior tire. So, and the division occurs like this. The division occur here, and then the division is and then oblique division. Oblique division means in the middle division is occurring. There is a middle division so that the spirally arranged. Spirally arranged means in the middle. This is actually 2-dimensional structure. We cannot understand it. But the 4 blastomere of upper tier, they do not lie over the corresponding blastomeres of the lower tier, but between them. So the upper is between and the lower is around them. This is spiral arrangement. What is rotational? Rotational means this is important one because it actually happens in humans. First division is meridional, vertical. Okay. The second division is out of the 2 cells which are formed, only 1 cell will divide first, which is equatorial, right? The second cell which is there, now it will go meridional division. One by one the cell is, forming. So the first 1 cell was there. After that, the 2 cells were there. After that, 1, 2 and 3 cells were formed. After that, now 4 cells are formed. What does it mean? 1, 2, 3, 4. One by one the cells are forming, and this is also in the rotational pattern. So it is actually the mammalian embryo are unusual because they have this type of division is known as asynchronous. What does it mean? All the blastomeres are not dividing at the same time. Blastomeres are dividing one by one. They are not dividing, so rotational is also there and asynchronous is also there. Asynchronous. So 3 types arrangement based on the arrangement of the blastomeres, either radial or spiral or rotational. In humans it is rotational. So the first cleavage was meridional, vertical. The second cleavage is rotational one. Why rotational? Because that was the first, that was the second. So first divide and it will form the 2 cells. The second will divide and it will form 2 cells like this. So 1 cell, then 2 cells are formed, then 1, 2 and 3 cells are formed, then 1, 2, 3, 4, one by one. It is not like pattern that all the blastomeres are dividing and 1 to 2, then 2 to 4, 4 to 8, not like this. Okay, so this is known as asynchronous. Now 3 types of classification of the cleavage we have studied. Here is the fair slide, you can see. Okay, so the various pattern: holoblastic, meroblastic. Complete cleavage is hollow, which is actually occurring in humans. Meroblastic, when cleavage furrow restricted to the germinal disc, yolk remains uncleaved. The second type, indeterminate, where the stem cells are formed in the earlier embryo, so that in case the embryo will be broken down, each cell is capable of forming complete organism, which is present in vertebrate. Determinate type, where the blastomeres have, since the beginning, they have their predetermined future. And if in case the zygote is broken down, embryo is broken down, then there will be, of course, a defect in the organism. Okay. The third one, that is the arrangement. Here you can see the first is radial, where the first division is right angle to the another one. Okay, and spiral, where the third cleavage, the 4th blastomeres, they are laid down obliquely so that the upper tier do not lie over the lower, but it is present inside them. So spiral arrangement is there. You can see here, once this one, then second, then third, these spirals are formed. Okay, now the next one, which is important for the human because it is rotational. The first division is meridional. After that, this one is 1 cell will divide, right? That is rotational. So 1 cell is dividing and then other 1 cell is dividing this way. So, and it is synchronous also because all the blastomeres are not dividing at the same time. Now we can write that human cleavage is holoblastic, yes, rotational, asynchronous and indeterminate. These are the 4 terms which we have studied so far. Okay, now we can see that we have already discussed here that after 24 to 30 hours, the first division occur, 2 cells, then the 4 cells, then 72 hours, 8 cell, and then the solid ball of mass. That is the 16 cell stage, which is known as morula stage. This 16 will go to the 32 cell stage, which is known as blastula. Now by this time it reaches to the uterus. Okay, because we all know that fertilization occurs in the oviduct. Now by the time the blastula is formed, this fertilized egg reaches to the uterus for attachment to the uterine wall. Now we have seen two stages, morula and blastula. What is the difference? Actually, the morula is mulberry. Morula word comes from mulberry, solid ball of mass which is 16 cells, okay, and it developed from zygote due to rapid mitotic cleavages. Blastula, the morula changes into blastula, and then this blastula form the blastocyst, where some cells go to the one pole and some cells surround the complete structure. So blastocyst, it has the two structures, that is the outer layer, which is trophoblast, and the inner cell mass. Of course, these are totipotent cells, and there is a cavity which is formed in between, which is known as blastocele. So a cavity which is known as blastocele. It is very much important for the point of board exams, because it is there in your practical also and theory class also. Okay, that generally question comes that draw the structure of blastocyst and write the functions of the different parts. You can see here. Now just here you can see the difference. Morula solid ball, this is hollow ball, and after that the blastula will form the blastocyst, where the inner cell mass will move to one pole, and you can see the structure here. When inner cell mass move to the one pole, that is known as embryonic pole, and abembryonic pole is the opposite pole to the embryonic. And from here, the inner cell mass, it is going to form the embryo. It is going to form the complete embryo. The cells which are just close to the trophoblast, the cells of that embryo proper or inner cell mass, these cells are known as cells of robbers, which are just close to the trophoblast. Trophoblast is the outermost layer. Now we are going to see the functions of different parts of this blastula. We all know that the cavity is blastocele, fluid-filled cavity, of course. And this blue layer you can see is zona pellucida. What does it mean? The size of the embryo is not increasing, only the number of cells is increasing because of this layer which is known as zona pellucida layer. It is still intact, and this makes the embryo non-sticky. The trophoblast is sticky layer. It can get stuck to the surface, but the zona pellucida prevent it from attachment. Zona pellucida is layer till the blastula stage is formed. Now outermost layer trophoblast. This trophoblast has the function to form extra embryonic membrane, and we will see the detail of all types of extra embryonic membrane in the coming slide. Extra embryonic membrane. For example, I can tell you that the placenta is like formed from trophoblast. Chorionic villi is formed from trophoblast, because these two terms are very common and you must have heard. Detail of all the different types of extra embryonic membrane we will going to study in the coming video. Now trophoblast, which forms the extra embryonic membrane, which takes part in the formation of placenta, and the inner cell mass, which actually forms the embryo. It is going to form the three germinal layers, and which will form the different organs. So now that's all for blastula stage. Okay. Now, very important event which confirms actually the pregnancy. Fertilization does not confirm pregnancy. The pregnancy is confirmed by attachment of blastula on the wall of uterus, which is known as implantation. And this occurs when the blastula reaches to the uterine wall. Now the trophoblast layer, it secretes proteases enzyme which digest the zona pellucida layer, and now the blastocyst coming out from zona pellucida. Zona pellucida, and by the sides in which the embryonic inner cell mass is present, it will going to attach with the endometrium wall. Okay, so digestion of zona pellucida is known as hatching of zona pellucida, and it is generally occurs by the 5th day, end of the 5th day after fertilization. Inability to hatch of zona will result in infertility, and premature hatching, of course, it will result in abnormal implantation. Why abnormal implantation? Because I told you that, yeah, this is the picture where the first cleavage, after that the second, then the fourth stage, then the morula. Now it has reached to the blastula. I told you that the zona hatching occurs at this time. If zona hatching occurs before, what will happen? This embryo, which has the trophoblast cell, because the trophoblast, trophoectoderm, they are very sticky, so they are going to stick on the wall of the oviduct, and the implantation may occur at the wall of the oviduct anywhere. That will result, of course, in the hemorrhage, bursting of that oviduct, of course, because as the embryo will develop, it will put the pressure on the wall of the oviduct, or the bleeding may occur. So abnormal pregnancy is the its reason, because of first reason may be ectopic pregnancy. Ectopic pregnancy means the implantation occur not in the uterus but anywhere else. Like, it's one way is implantation occurs if premature hatching of zona pellucida will be there, then the implantation will occur in the oviduct. Okay, so yeah, this is actually the structure of blastocyst. We have already done embryonic pole and embryonic pole. Here is the cavity. Zona pellucida already degenerate, and blastocyst takes the nourishment from uterine secretion. Yes, now it start to. So I have just told you that the zona hatching, when the blastocyst is coming out after, this is the trophoblast layer, and this is the zona pellucida layer. This is actually the inner cell mass. At this time, when the embryo is coming out, it is actually come, it is said that it is actually come in the form of 8, because it has to come out. So it is coming in the form of 8 shape, and it can be broken down into 2 or more parts. And because the human cleavage is indeterminate, the blastomeres, earlier blastomeres, they do not indeterminate. Earlier blastomeres, they do not have predetermined future. So in case it is broken down into 2 parts, then each can form complete human. Because it is these 2 humans are formed from one embryo only, it is known as, these are known as identical twins. Yes, that is how the identical twins are formed. Of course, they will be formed from one zygote, so the same sex, and their genotype, DNA will be same. No other 2 organisms have the same DNA, only the identical twins. So DNA is same and of same sex. You cannot even identify. You must have seen identical twins in your classes also. Non-identical. If in case the 2 sperms will fertilize 2 eggs, so 2 sperms and 2 eggs meet. Because sometime we have already discussed that from one of the ovary in 1 month, 1 egg is released. So from the right ovary, in case there is a delay in egg ovulation, and these 2 eggs which are formed from both the ovaries, maybe they are close to each other, then the 2 sperms can fertilize 2 different eggs. Then they are known as non-identical. The sexes may be different, maybe different, maybe same. Depends on at the time of fertilization. So sex may be different. may be same, may be different. And of course, two zygotes are there, so DNA is not same. So non-identical twins, which are also known as fraternal twins, right? Fraternal twins. So yeah, we can see the picture. Identical twins are formed from same egg. Two brothers, two sisters, right? From same zygote. And non-identical fraternal, non-identical twins are formed from two different eggs. Okay. Yeah, this is the picture, ectopic pregnancy, because here the zona premature, zona hatching, and ectopic pregnancy in the fallopian tube. So that's all for today's class. Thank you so much, children. Now in the coming video, I'll start with the after implantation, that is the gastrulation. From there we will start formation of 3 germinal layers and fate of these 3 germinal layers. Thank you.